Good evening, dear friends. I welcome you back to our channel. Today we will discuss classification of viruses. And the question we will ask ourselves now say, how would they take classify viruses? Wetin be the methods, wetin be the criteria we would they use classify viruses? This one dey important. Now we don't do many videos on viruses and we don't talk say when you call something a virus, wetin dey for there now, we get wetin they call viral DNA or RNA, and so we refer to them as viral genome, and that viral genome go dey covered by something wey we dey call capsid. That capsid na protein coat, and this one dey very important to note. Now we realize say our viruses, we dey classify them in two ways. We get two major ways wey we dey take classify viruses. We fit classify viruses on the basis of wetin we dey call morphology, and morphology na big science term to mean shape or size of something. So we fit classify viruses on the basis of their morphology. Another way wey dey take classify viruses na on the basis of the kind of genetic material wey the virus dey carry, whether na RNA or DNA. This na another way wey we dey take classify viruses. Now if we go back to our discuss on classifying viruses on the basis of morphology, we go come dey look say okay, e get two major classes wey viruses get if you dey classify viruses based on morphology. And the two major classes na say virus dey either get helical symmetry or they will get wetin we dey call icosahedral symmetry. Now wetin these two things be? If you remember, when we talk about helical symmetry, if you remember, we don discuss DNA, and we know say the structure of DNA na like something wey we dey call helix, and that helix na say e dey intertwined together, and na wetin the DNA structure bin look like. So many times, if protein coat of virus bin get this structure wey be say e dey intertwined together like that all around the viral genome, we dey refer to that as the helical symmetry. E get many types of viruses wey dey get all this kind of symmetry. Now we need to remember say our capsid dey made up of wetin we dey call protomeres, and these protomeres, wetin dem be? Dem be subunits, small units of proteins. So if those proteins join together, those small units, they will form protomeres, and those protomeres, if they join together come form this helical structure, they go round about the DNA or the RNA of that virus, and this one na wetin we dey call virus wey get helical symmetry. So one dey important to note. Another important thing na the other type wey be wetin we dey call icosahedral symmetry. Now this one dey complex. E get wetin we dey call complex polyhedron, and wetin we mean na say this icosahedral shape now e dey get faces, about 20 faces, and those 20 faces na face of equilateral triangle. We know equilateral triangle, this one na triangle wey get all the length na the same. So these viruses, dem bin dey, and e dey important to understand them. Now this icosahedral shape, e will get 20 faces of an equilateral triangle. E will come get 12 points, 12 different corners, and na this complex shape some viruses dey get. Examples of viruses wey get this kind shape na our herpes simplex virus and our HPV, that's human papilloma virus. All these ones na examples of viruses wey get icosahedral shape. Now viruses too, they also classify viruses on the basis of the type of genetic content wey the virus get. On this basis, we dey divide viruses into two. E fit be DNA virus or RNA virus. This one dey important to note. And we come realize for science say RNA virus dem plenty pass DNA virus. RNA virus dey take up almost 70% of all the viruses wey we know, while DNA virus na just about 30%. Now examples of our RNA virus bin include viruses such as our Ebola virus, SARS virus, polio virus, HIV virus. All these viruses dem be RNA virus. Meanwhile, the DNA virus include viruses such as our smallpox, adenovirus, and herpes simplex virus. All these viruses dem be DNA virus. Now e dey important to note say most viruses dey inert, meaning say they dey inactive once they dey for the environment. They dey only function if dem dey inside a living cell. Now in fact, if dem dey for environment, many viruses dem dey crystallize. They no dey function at all until they find their way into a living cell. Now e dey important to know say viruses na wetin we dey call obligate intracellular parasites. Obligate intracellular parasite. Wetin this one mean? Obligate mean say if they no dey inside living host, they no fit do anything. And the intracellular means say they will dey inside cells, they go dey live for inside cells, they reproduce for inside cells, and then be parasites. This na wetin viruses be. Now the last thing to note na say many viruses as dem dey, the capsid wey be the protein coat of those viruses, na the viral DNA or RNA wey dey for inside the virus na im dey code for all those proteins wey bin dey surround the viruses. So thank you very much for lis ten ing to this lecture. Abeg, make una like our con ten t, share our con ten t, share am for una friends, just to make your science better. Have a wonderful day.